So I'm going to speak about data science in biophysics for applications in drug discovery, and of course I'm going to mention also the AI component. So this is going to be more of a technical talk regarding biophysics and how we can use it in precision medicine. So we'll start with precision medicine and explaining what's the connection of biophysics and precision medicine. And then I will move on to biophysics data sources, how we can utilize this data for artificial intelligence, and in turn, how we can use data science, artificial intelligence in biophysics for drug discovery, which is the important thing for humans. And then I will mention an application from our lab, a specific application on how we use artificial intelligence to predict protein membrane interactions, and I will explain why that's important. And I will close with the applications in drug discovery and some concluding remarks. So what is precision medicine? So basically this concept of precision medicine, which is quite new, it's here just for the last 15, 20 years, is the ability to offer the right drug to the right patient for the right disease at the right time and at the right dosage. So we want to give patients the best possible medicines. And genetic and metabolic data now allow these drugs to be specifically tailored for specific patient groups. I will give you a specific example why that's important. An example, for example, in chemotherapy. Before, all cancers were just treated with cisplatin, so one dose and one medicine would treat all cancers. But we now know that that's not the case. Essentially, the tumors have their own DNA. They have mutations that cause the tumor genesis. And the specific tumor profile of the specific patient and with a specific cancer is very different from others. So we want to give patients specific treatments. And not only that, also in the dosage. So before we would give one dose for all people. Some people would respond to a normal dose. Some people would respond to a lower dose or higher dose. And some, as mentioned, would respond to completely different medications. So for example, it depends on how people metabolize drugs. For example, I cannot drink coffee. It's very difficult. Like one sip for me is too much already because I am a low metabolizer of caffeine. So I don't metabolize caffeine. So I respond to a very low dose of caffeine. So that depends on my own metabolic profile. And now with the data of each individual, we can tailor doses and we can tailor different treatments. And for those in the audience that are not biologists, I have this slide showing that we have the cell. And in the cell, we have this nucleus, and then inside we have our DNA folded in chromosomes with this characteristic shape. And the chromosomes are nothing but folded DNA, 2 meters actually, if you unfold it. And some parts of the DNA are coded in genes. These genes are transcribed by the RNA in the central dogma of biology. It's being brought, the information of the gene is being brought to the ribosome. Then the gene, essentially information is translated into amino acids, into proteins. And these proteins, when they exit the ribosome, they exit in just a straight line. And then they fold in 3-dimensional structures, which we can visualize using X-rays. And why is that important? This small entity here called protein. It's actually really important because the proteins are those that do all the functions in our body. So my ability to speak, to move my hands, it's all coming from different proteins that communicate with each other and give signals. And these proteins, they have these 3-dimensional structures in space. So you can see here, this is a virus, so many different proteins coming together, and the individual proteins are listed here. And it's the structure of this protein, so having this 3-dimensional information is extremely important because proteins are pharmaceutical targets. Proteins are the entities that we use to create drugs. And I'm showing 3 viral examples here, examples from proteins of viruses. This is from HIV, the reverse transcriptase. This is from the influenza virus, the M2 transmembrane membrane protein, and the NS5B of the hepatitis C virus. These three proteins are completely essential for viral replication. So what is a drug? It's a small molecule that we place inside the protein and stop the virus from replicating. Because if the virus needs this protein to replicate and we stop the protein, then the virus stops replicating, so we have a cure. So the premise of drug discovery is actually that you create these small molecules that are, you can see here. You place them in these pharmaceutical targets, and then you proceed with drug discovery. So that's exactly what I said. So drugs basically block or activate disease proteins. For example, the proteins that cause pain, that cause the signal of pain, we can block them with paracetamol, Depon, I'm sure you all know it, or aspirin. So you can see it here. So they're very seemingly simple molecules, but they have a huge effects in our lives for therapy, relief, prevention, and actually they've improved a lot our life, the quality of life, and have prolonged our life expectancy as well. And now more specifically, now utilizing this concept of blocking these proteins in precision medicine. So let's take a patient population of that has lung cancer. We can actually differentiate these lung cancer patients in different groups. And I'm showing here just one of these groups, where with genotyping, it was found that 4% of the patients have a rearrangement in a specific protein, anaplastic lymphoma kinase, just in cancer and just in these 4% of patients, that results in carcinogenesis. So then what we can do is to take this protein, to visualize it, see it in three-dimensional space, and design a drug for this specific subset of patients. And this is how crizotinib, which you see here in green, was created. This molecule just binds specifically for anaplastic lymphoma kinase patients. So when a patient goes into the doctor and has cancer, will the doctor prescribe immediately crizotinib for the patients? No, they have to do genotyping. You have to see if they have this rearrangement in ALK protein. If they don't, then even if a patient has lung cancer, they won't respond to crizotinib. They need to have this rearrangement. But that's a very, very targeted approach. So this means that you don't have the side effects of all these chemotherapies that target these multiplying cells, and then you lose your hair and all these devastating side effects. This is a very specific targeted approach for only these 4% of patients. So that's called precision medicine because you target precisely those subgroup populations. Sorry, I have some Greek here. So in the phases of pharmaceutical development, that's quite important, especially for preclinical trials that start with a target discovery. And once we have this target, so in this case, it was the anaplastic lymphoma kinase. So here you need the target discovery. And when you have the target, which is usually a protein, you go on with a drug discovery and eventually going also in humans. So this is a very, very lengthy process and very, very costly. And we think that with AI and other approaches that are more rational, we can make this cost less and bring drugs faster. So basically, why is this so costly and so it takes so long? Because usually in the past, the pharma industry didn't have any informed approaches. They just use these high throughput screening methods. So basically, they're screening hundreds of thousands of molecules with no information, so they're not informed decisions, just randomly testing against a pathogen to find a molecule that is potentially a drug. However, if you do this random approach, the estimated of small molecules that can act as drug is a huge number, and it's actually much more than the atoms in the world. So even if we wanted to try all possible combinations of molecules for disease indication, we wouldn't have enough atoms in the world to synthesize these molecules. So it's not possible to do random discoveries or high throughput screening if we want to be efficient. So I'm going to discuss how physics-based and artificial intelligence approaches can lead to targeted therapies. So with these approaches, basically as mentioned, we identify the important gene for a disease, which is usually a protein, pharmaceutical target with small molecules, and then we design a candidate drug, especially for the specific protein structure that we have. And that's now possible. It wasn't in the past because it's quite difficult to get these three-dimensional shapes of the proteins. But we have a recent data explosion in medicine and biology and biophysics. In terms of drug discovery, artificial intelligence can also make significant contributions for the choice of the pharmaceutical target, which is the protein that we're going to use for drug discovery, to discover where a drug will bind, to design the bioactive compound, to predict its toxicology and its selectivity, so side effects, to predict how the organic chemist will synthesize this compound, and to optimize this compound to have the best pharmacological profile for humans. So before AI came, we used to have traditional computational biophysics approaches. So I'm going to describe how I have witnessed it. So before we had all this data, because these AI approaches are not so new. Actually, we know them for 50 years, since 50 years, but it's only now that we can use them because now we have the data. So before these AI approaches, we have the traditional computational biophysics, where basically you have rules. For example, Newtonian mechanics. We know how a system would work, would move with Newton's law of motion. And we also have inputs. For example, the coordinates of the three-dimensional structure of the protein. So using the coordinates of the protein and Newton's mechanics, you can actually do a simulation to have the protein move in real space and time and observe its output. So how the protein will propagate in time, how it will move. With machine learning, we have a slightly different, or completely different approach, not even slightly. So here, we don't have actually rules. We only have inputs and sometimes also outputs. So we have all the data. So we only have data. Then we combine this data in statistical models, in pattern recognition, for example, and others, and the algorithm will create the rule. So instead of giving the rule as an input, the rule now becomes the output. So basically, with machine learning, you combine the data with statistical models. The algorithm makes some correlations of the data that you are giving it, and then it creates rules and predictions. And we can classify it in these two classes. Supervised learning, the computer is trained on data inputs and outputs with a goal of learning a general rule that maps the given inputs to the given outputs. So that's supervised learning, when you have both inputs and outputs as the input. And in unsupervised learning, you don't have the outputs. You only work with the inputs and you discover the pattern of a structure in the input. So these two different classes, supervised machine learning, these are trained on data when we know the output, as mentioned, and we can classify them in two different schemes. So in classification, you want to predict which category the data will be in. So for example, I'm showing an example of the petal width and the petal length. And here we can classify those that are of a petal morphology of virginica and petal morphology of versicolor. So we just classify two different classes. And in the regression, we fit the data. So basically we predict a numerical value based on previously observed data. So it's just a fitting of the data. And an example of supervised learning with classification for drug discovery, we create a data set of inputs and outputs. So basically we know which molecules are active and which molecules are inactive. And we also know we can create some models that are called pharmacophores that are active. So we create labels for which of these data is active and which is inactive. Then we train the algorithm on the active and the non-active molecules. And the result is a classification on this is going to be on unknown structures. This is going to be an active versus this is going to be a non-active. So this is just a supervised learning with classification, so with labeling the data in inactive and active. Now in unsupervised machine learning, here you don't have the outputs. You only have inputs. So you fit the data. So here you have, for example, as inputs, the one molecule. Basically, whatever this molecule has as descriptors, the bond lengths, the charges, the position, the coordinates. So you input these, an example of unsupervised machine learning in a neural network. You provide these descriptors as input, and then you ask this network to create associations between the input data that you have given it. So these hidden neural layers will compute a weighted sum of these input values, taking into account all the parameters that you have given, and with an activation function, it will calculate the output result, which is, in this case, is the quantum mechanical properties. So now, as mentioned, with the data that we have, we can use these approaches to predict new compounds and to predict different aspects of proteins. And I will show you some example from our lab. I will start with some work that we were doing before we had the data, so before AI. And then I will show you how we continued using these AI algorithms. So 50% of pharmacological targets are membrane proteins. So basically they are proteins that contact the membrane. So here I'm showing a protein which is on top of the membrane. And here I'm showing you a protein that's inside a membrane. So these are very important proteins because half of these proteins that are in contact with a membrane are a pharmacological target. So that's quite important. So, however, this protein-membrane interface, where exactly the membrane is, we don't really have this information because this structural data, they cannot really visualize the membrane because the membrane is very flexible and moves. Usually when you see a protein structure, you don't have the membrane together. So you don't really know where the membrane is. But that's really important because they are in contact with the membrane. So if you want to design better drugs, you better know where your membrane is. So knowing where the protein-membrane interaction occurs is a new drug design strategy, which we didn't really have access to because we didn't really have the data to know where the membrane would be. So we created a workflow to identify where this protein-membrane interface is and to use it in drug discovery. So first, we study dynamics of proteins to see how it moves in space and time. We use machine learning to understand where the membrane is. Then we identify where a drug molecule could bind, and we see whether this molecule has an effect on the overall protein structure. And then we go on with a drug design strategy on these druggable sites to find molecules that would actually go and fit into this cavity, and then we test them, of course, experimentally. So we did a lot of studies first on pure membranes to understand how membranes work. So I'm showing here some of our publications and how we could reproduce in the computers with computer simulations of membranes. And here I'm showing such a simulation where you put randomly water and the lipids, and then you spontaneously see in the computer the formation of these lipid bilayers. So the membrane forms spontaneously. And we also had, can we play this movie on the left? I don't know, can you hear me? Can you play the movie on the left, or can I do it from here?
Yes.
Ah, okay. Yes. And so in terms of dynamics, we can also monitor how the drug really explores a protein surface and how they can find the cavities. And we can also explore how a membrane protein is moving inside the membrane in the presence of the drug. So we can do all these simulations with these physics-based approaches, basically using very, very powerful supercomputers, because these are simulations that are quite difficult to do. And we use this strategy on a lipid kinase called PI3 kinase alpha. This is the most mutated kinase in human cancers. And it actually has two hotspot mutations, which you can see here, glutamic acid becoming a lysine and a histidine becoming an arginine. And these are actually 30% of the breast cancer population. So you can imagine how important this drug target is. If you remember in the lung cancer example, we had only 4%, and here we have 30%. So if we manage to find a therapeutic strategy to understand how this kinase works and to design better therapies, it would really benefit patient populations. So we did all the simulations, and we kind of understood how the protein is binding on the membrane and how other regulatory elements work. I won't go into the details. And we also did the drug design approach I showed you to design these molecules in the pockets that we found. And we have data that shows that these molecules were actually successful. So they were allosteric inhibitors, so they were inhibiting the activity of the kinase. So I won't go in the details here, but I just want to show you that it was selective for PI3 kinase. So in the concept of precision medicine, that we want to target the PI3 kinase mutation, the patient populations, we designed molecules just for this cancer type. And here you can see the tumor volume for PI3 kinase wild-type tumors, so tumors, breast cancer that is not dependent on this mutation. So when you give our molecule, here in light gray, nothing happens to the tumor, so the tumor really grows. But when you give the specific drug to the tumor that is mutated of PI3 kinase, you see that the tumor is really affected with respect to the control. And we could also show it in genetically modified mice. So this approach, which is, I mean, usually we didn't use any eye for that, is kind of successful. And we could also show that our protein, so PI3 kinase, could attach onto the membrane. So we could understand how the protein attaches on the membrane. So using experiments and simulations. And that approach, this physics-based approach, was also successful in another case, the angiotensin 1 receptor, where we could show that in this protein, there is a molecule of cholesterol binding right here. So there is a binding pocket right here inside the membrane. So you can use this strategy inside the membrane as well, which was very important for drug discovery. And on to another example, again with physics, so no AI so far, so I will come to that. 30% of human tumors, another very important protein, KRAS, in collaboration with Novartis Institute of Biomedical Research in Boston. We wanted to study how this protein dimerizes, because this interaction with a membrane and with this other kinase is very important. But no one knows really how this complex is forming. No one knows how it interacts with a membrane. No one knows where is this other kinase. So we really don't have any information. And again, using these, not AI, but physics-based problems, we try to understand how the protein dimerizes and how it attaches to the membrane. And of course, the first thing we did before trying to use these physics-based methods, because they're very expensive and the recurrent supercomputer was to try this AI approach, AlphaFold, that I showed you, because I showed you that AlphaFold cannot predict the structures of proteins where you don't have the information from experiments. Unfortunately, it didn't work, so it gave us these kind of complexes that are non-physical. And the reason why it didn't work is that this is a very complex problem. It's not like the protein folding problem where you have the string of amino acids and you fold it in three-dimensional. You now have two proteins that come together, and there's also the membrane. So AlphaFold could not predict it because it didn't have information about the membrane, and it also didn't have information about how two proteins come together. So yes, it's useful to predict one protein on its own, but it cannot predict more complex systems closer to the native environment. So when AI failed, we went back to our physics-based methods. We retrieved two structures from crystallography and NMR. And we started to simulate this again in the computer, like with the dynamics, as I showed you, and understanding how KRAS could potentially dimerize in the presence and absence of this other protein. And we did find, indeed, some significant results that showed that, indeed, this other protein stabilizes the dimer, and there's some interactions. But, however, we didn't really know, again, if this one structure that we're simulating is enough. So we tried other methods. This is another simulation method. But again, we only had this very, very limited understanding of how the proteins dimerized. So we again tried even more to understand how this dimer is forming with other approaches, basically going into simpler representations of the system, and with providing some energy to the system to move. And here I'm showing another video of how this works. So by giving energy to the system and simplifying it, now we can see spontaneously how the two proteins can dimerize. And to try and test if this is correct, we took some examples of the literature. So insulin, a dimer, Barney's bar star, RAS-RAF, the transmembrane domain of EGFR, and rhodopsin. Here is rhodopsin. And we tried the strategy, and actually we could see that, yes, indeed, these proteins are coming and going, and we can now map the full process. We're not limited to one very specific view, but we can see exactly how they come together. And we built these three-dimensional maps of what are the most stable structures. And these are these maps here. So basically, the very dark spots are the stable conformations of the protein. And indeed, these conformations are the crystal structures. So some of them, not all, but some are the crystal structures. So this method showed that we can use it, in fact, to predict this dimerization. Okay, I don't have KRAS here. Okay, so we used it also for KRAS. I don't have the simulation slide here, but it doesn't matter. And we could also observe this folding and this coming and going of the KRAS dimer. But this really took us, I don't know, 3-4 years to do. And so this was really not efficient. And since we had artificial intelligence now in our hands and the data, we decided to go on and study the membrane binding domain identification using machine learning. So where KRAS, this protein, binds on the membrane, we could now use it using machine learning to identify where the protein will bind on the membrane. So I'm going to describe now how we used maybe 3 years to do the simulations that I showed you, but now after designing this machine learning strategy, we can predict this protein membrane binding in just a few minutes. So we turned, as mentioned, to AI to predict these membrane-protein interactions. So the hypothesis is that we want to predict where the amino acids bind on the membrane. For that, the most important thing is the data. So if you have data that is correctly annotated and it's a lot of data, you can ask this hypothesis and you can test it. If you don't have enough data, it's not possible to do it. So we have data for peripheral membrane proteins with experimentally known membrane-penetrating amino acids. We extracted some features of proteins that we know. What are the features? What is the feature of a protein that goes onto the membrane and of the amino acids? And then we use the supervised learning that I showed you in the beginning to classify whether an amino acid is protein membrane interacting or whether it's not interacting with the membrane based on the features. And then we evaluated the predictive model based on an independent test set. So let's see how this approach works. So again, we started with a data set, we prepared it, feature extraction, and machine learning. We took 54 peripheral membrane proteins from the protein data bank with experimentally known membrane-penetrating amino acids. We prepared the data set by deleting some parts of the molecules that are not interested and other preparation. We generated descriptors for the amino acids that are contacting the cell membrane, and then we did machine learning. For the data selection, again, these 54 peripheral membrane proteins have about 15,000 amino acids. We split them in validation set and training set. So we have 46 membranes in the training set. Then we remove amino acids which are inside the protein, because only outside the protein will contact the membrane, so 2.5 angstroms. We also remove non-hydrophobic amino acids. We do also for the validation set. And also we have an independent test set of 11 peripheral membrane proteins to test our predictions. And the percentage identity between all sequences is less than 40%, so that we have an unbiased data set. So the first thing to do is to calculate which features are important for amino acids that contact the cell membrane. And this feature importance can be calculated by algorithms. Here we used these ensemble tree-based algorithms. For example, the extra trace classifier and the light GPM classifier. And this showed us what are the important features. And what we found was that they are the hydrophobicity, solvent accessibility, secondary structures. But we also found that the evolutionary conservation score, flexibility, and the hydral angles were also important. Then we proceeded to label the data set, separating the amino acids in two classes, the ones that penetrate the membrane and the ones that don't penetrate it. And this is our training set, but you can see that the penetrating original data here is much more less than the amino acids that don't contact the membrane. So you have the problem of imbalanced classes. And that's very important in machine learning, because if you have two classes that are imbalanced, it's going to be very difficult to do accurate predictions, because you will have imbalance between one class versus the other class. So we need to create balanced classes using different algorithms. For example, we can oversample the minority class, the membrane penetrating amino acids, using, for example, SMOTE, or undersample the majority class. So we actually did that, 6 different approaches. First with undersampling the majority class with these different algorithms, oversampling the minority, and also using class weights to say to the algorithm that the membrane penetrating amino acids have more weight. So we use these six different approaches for balancing the data set. And the next step is to choose which algorithm you're going to use for classification. So this is a classification problem. We want the membrane penetrating amino acids versus the ones that don't penetrate. So we have a classification problem, and you have so many different algorithms these days in the literature, so you really don't know which one to use. So you have to try many different algorithms, because every classifier works in a different manner. It has the potential to predict correct samples, but others cannot. And you don't know a priori which one is the best classifier for your specific problem. So here we used many, as I will show, we used an ensemble approach. So we used many different classifiers to eventually decide which one was the best for our problem. After the classifier choice, you need to do a hyperparameter optimization. So these classifiers, they have their own parameters. And depending on the parameters of these classifiers, the model may underfit the data, it may overfit the data, which you don't want, neither of these cases, or you can have a good and robust prediction. So this hyperparameter tuning basically is training thousands of models with different parameters for each classifier to find which one will be give the good and robust model. And you keep the parameters of the model with the best performance. So these are the different steps. To do this hyperparameter optimization, starting with a training set, one approach, there's others, but we used the 5-fold cross-validation. So we split the data set in 5 folds. These folds were balanced 6 times with this oversampling and undersampling. So we have 5 folds, but actually 6 data sets that come from oversampling and undersampling. And then we use this 5-fold cross-validation to find the best hyperparameter optimization scheme. Yes. So now we have to choose with supervised learning to train the ML algorithm and choose the best classifier. So to train and validate this, we chose 21 machine learning classifiers. I showed you the 5-fold cross-validation. And here, basically, we train hundreds of thousands of models because we have the 21 machine learning classifiers, and for every classifier, we have the 6 different training sets, and for these 6 different training sets and the 21 machine learning classifiers, we have thousands of hyperparameter optimizations. So you need really powerful machines to do this because you really need hundreds of thousands of models. Here are the 21 classifiers that we chose. And as mentioned, we don't know which one would be the best one. So what we did was to actually create a voting scheme. So we took, yes, it's here. So we took a meta classifier. So when training all these hundreds of thousands of models, in the end, we use a meta classifier, which is basically a voting classifier, which will vote which one was the best model. So, I mean, we didn't use it manually. And the metrics that the meta classifier uses is the recall, the precision, and the F1 score. Okay, spoke the hyperparameter optimization. Yes, these are our metrics based on true positives, false negatives, and true negatives. So the algorithm finds what are the actual true positives that they are true, so they're penetrating the membrane. So these are your true predictions. Then you have the false negatives, so predictions that are shown negative by the algorithm, but actually they are positive experimentally. False positives, ones that are predicted to be correct, but they are not. And true negatives, so they're not penetrating the membrane, it's true experimentally and computationally. So based on this confusion matrix, this we call confusion matrix, you have different schemes of calculating these different metrics that you use to evaluate the performance of the models. So as mentioned, we had these hundreds of thousands of models, and we use this voting classifier. The majority classifier voting for the first level classifiers is 21 classifiers. We also had a stacking classifier to train a classifier based on the output of these of the best performing classifiers, take all possible combination, and test which one was the best on the validation set, which we separated in the beginning. So that's quite important, split into training and validation sets. So the results of the meta-classifier was that you have an F1 score of 0.86, an MCC of 0.71. It's not the best, but it was actually quite good. It showed that we have predictive value. So the best here would be 100%, meaning. And the voting classifier told us that these 5 algorithms, linear discriminant analysis, logistic regression, linear SVMs, and decision tree classifier in the light GBMs were the best. So let's see some of the predictions. How does it look? Here with red, you can see the putative membrane plane. With red, you see the experimentally known membrane-penetrating amino acids, which are predicted from our tool. These are the true positives. Experimentally known membrane-penetrating amino acids not predicted are with green. These are the false negatives. And false positives are amino acids predicted from our tool that are not experimentally proven. So when we observed this, we found that actually we have these purple amino acids that are false positive, but actually it's clear that some of them, they're not false positive. I mean, this cannot be a false positive. It's actually a true positive, but it was not determined in experiments for some reasons. But it's actually true positive. So once you go back and do manual inspections of these false positive predictions, you find that actually 2/3 of the false positive predictions are in fact correct predictions, but they're not proven by experiments. So when you include them, then you increase the prediction from 0.86 to 0.92. Then we compare with other models in the literature. For example, here is our model with a membrane plane with the correct predictions, and you see here another tool that's completely misaligning the membrane plane. And here another tool that has a prediction, I don't know, somewhere else. So it was clearly outperforming the competitors. And also in completely independent tests that we collected afterwards, you can still see that it's doing very well to predict this interface. And further inspecting some of the false positive predictions that were not in the membrane plane, we found, for example, that here the algorithm found an amino acid that it predicts it goes to the membrane. It doesn't go to the membrane, so this is false. But when you visualize the actual structure, the biological assembly of the protein, you see that it's actually a tetramer. So there's 4 subunits coming in together. So here, where our algorithm predicted an amino acid, it's because another protein comes in. So it also has some predictive value for protein-protein interactions. And in terms of AlphaFold, if you want to predict where a membrane will bind on the protein, you have to take care to use only regions of amino acids with AlphaFold that have very high fidelity. These are the blue regions. You see here these completely disordered regions. It means that AlphaFold cannot really find the structure, and they are colored in red because there is not a high fidelity score. So you have to take care to include only the blue regions, because if you use, for example, this structure to predict where the membrane is, clearly there's no result, it's wrong. But if you use only the high fidelity region, you predict that this is a transmembrane domain. And so there needs to be caution when using AlphaFold predictions, because you need to know which ones is going to be correct. And yes, so our algorithm also predicts the transmembrane domain, so inside the membrane, although it was not trained for proteins that are inside the membrane, but only for those that are on the surface of the cell membrane. But still, it can predict it with quite good accuracy. In the end, we created a web server where you can input your structure, and the algorithm, when you input the coordinates, will tell you where the membrane amino acids are. And the algorithm, as a web server, it also has drug discovery capability to detect where possible druggable pockets exist, where you can do drug design. After we published this paper in the web server, we had 2 prospective applications coming in and using our DREAM. So this is not my figure, this is a figure from the Sabatini group in MIT. So they had a protein and they didn't know where the protein binds on the membrane. So basically they took their protein structure and they made predictions with our web server, and they found these 3 different spots where it contacts the membrane, and they then did experiments. They did experiments, and the experiments showed that the DREAM predictions were good, so we were really happy to find that you can use it also prospectively. So people used it independently. They confirmed which are the protein membrane amino acids with DREAM, and then they did experiments, and the prediction was correct. And that's another study after we published the algorithm. And so our membrane binding, okay, yes. So I will then skip the drug discovery part to go, because I only have 2 minutes, to go finally into just for 2 minutes to show you how we can apply natural language processing to proteins and to the specific problem of protein membrane binding. We can use natural language processing as the algorithm. You all know ChatGPT, right? This is based on natural language processing. And actually, this natural language processing can be used on proteins. Protein sequences consist of 20 amino acid letters, similar to an alphabet. So protein sequences can be used as sentences and their amino acids as words. So we can use the entire protein sequence as a new language. And this has been successfully used, these language models, to predict, again, protein folding, not with structure, but just with a sequence. And the advantage is, that's from Facebook, the other one was from Google. The advantage here is that the AlphaFold takes minutes or hours, hours or days. This takes seconds or minutes. It's very, very fast. And these protein language models in biology, they come from these databases, protein databases that are created from Facebook, and this is from Google. So here we use these protein language models, again with a data set. We constructed features of the words, so of the amino acids. We trained the machine learning classifiers, and I'm going to show you the results. In 2020, end of 2022 to 2023, there is this new data set with many more structures than what we had previously. So we could use this deep learning approach for these 2 different data sets. We constructed the embeddings. This is the final data set, labeling amino acids as membrane penetrating and non-membrane penetrating. Splitting again the data set. And finally, training and the results. So the results of the protein language model applied to protein membrane amino acid prediction was very good. So we could actually predict with high accuracy and just in seconds. This is in seconds instead of minutes that DREAM took. The same essentially predictions with DREAM. So it was quite successful. But the only limitation here is that if your data set does not, because the data set that we had was for 9 protein families, if we gave it a different protein family, then it didn't predict anything. So to conclude, this is my final slide. AI and ML needs big data. In this case, for example, with a natural language processing, we didn't have data for all protein families. We only had for 9 protein families. And so data needs to be of high quality and correctly annotated and unbiased. Otherwise, you're not going to get any result. You're going to do overfitting for these, for example, 9 protein families. AI cannot predict a property if it's not in the training set. If the AI has not seen it, it cannot predict it. Like for the example of AlphaFold, if it doesn't have the information about the membrane, it cannot predict it. So it's useful because it can give you correlations of data that you cannot predict with a human eye, but if it's not in the training set, you cannot really predict it. So molecular simulations and these physics-based methods that I showed you in the beginning are of value because they provide these physical insights. And AI and these physics-based methods are complementary and can be used in tandem. And we also have a special issue on machine learning in biochemoinformatics in the Journal of Chemical Information and Modeling. And I would like to close with acknowledgements of the group, of the funding, of you for your attention, and the organizers for the kind invitation.